Okay, team, here we are back again. And we are in the throes of talking about the division Ascomycota, which is the second division of fungi that we're covering. We've covered some of this, and so we'll have a little review. We talked about important edibles, morels, and truffles. And we haven't talked about these other topics. There's the morel, which we discussed, and there are some truffles. So it's a good time, I think, to talk a little bit about the life cycle of the Ascomycota. We can look at this next slide and kind of use this as our basis for going a little bit through the life cycle. Now, we did the Zygomycota life cycle in detail, and that's one that you could expect to see again. We will not do quite as much detail on the Ascomycota, and then when we get to the Basidiomycota, we'll do a full-on life cycle for that group. But one of the common names of the Ascomycota is the cup fungi. And you can see this is the fruiting body of a species or a genus called Peziza. And you may have seen, probably not this particular species, but one similar. If you walk around in the woods, especially in the late winter, you might see these fruiting on twigs on the forest floor. What puts the particular fungus in the Ascomycota is the formation of a particular structure during sexual reproduction called the ascus. And we can talk about what that looks like. An ascus is shaped like this, like a finger almost. And inside are 8 ascospores. 1, 2, 3, 4, 5, 6, 7, 8. So this is an ascus with 8 ascospores. And if you have a fungus that makes an ascus with its 8 ascospores, then we put it in the Ascomycota. That's what puts it in there. The yeasts are in this group. Although they are unicellular, they do have sex. And when they have sex, they make an ascus with 8 ascospores. And so, that's why we often ally or why the yeasts are put in the Ascomycota. Okay. So you might think about a little more about the life cycle for just a second. If we have meiosis occurring, we get 4 cells. So the question is, how do we get 8 cells? What's the significance of that? Well, what literally happens is meiosis occurs and you would get 4. 1, 2, 3, 4 as a product of meiosis. And then each of those 4 does 1 mitotic division to give you your 8. So if each of the 4 cells does mitosis 1 time, you get 4 more, and that's how you get the ascus. Okay, that's about as much as we'll say about the life cycle of this group. Okay. Let me get rid of this. And on to the next slide. And this is a fascinating little topic that we're going to do here now. There is a fungus called Claviceps purpurea, also known as the ergot fungus. And it's important to understand what you're looking at here. What you're seeing is wheat or rye. I think, oops, darn, I've done it again. Oh gosh, how many times am I going to do that? I ask. It happens. Need to remember to annotate. Okay, there we go. Now it won't happen. Okay, what you're looking at is rye, which is a grain, or it could be wheat. It's hard to say, but I'm going to guess that this is rye. And so you have these little heads of rye. What you're seeing here is the claviceps, the fungus. And the structure is a spore-producing structure. And so literally what's happened in this case is a spore has come and landed on one of the grains, one of the kernels of the rye, and germinated and begun to grow. And that's what you see, these little kind of rat turd-like structures that are present are the actual fungus, which we call Claviceps purpurea. Purpurea because they tend to have kind of a purplish color. It's a little hard to see in this picture. So to be clear, this fungus is actually a parasite of grain. It feeds upon the starch in the grain kernel, and then it makes its spores and continues its life cycle. However, if this fungus is ingested by mammals, it causes an illness, and that illness is referred to as ergotism. So that's why this is commonly called the ergot fungus. So if we start thinking about ergotism and some of the symptoms, lots of physical symptoms. It affects both the kidney and the liver. The toxic alkaloids in the ergot are filtered out by the kidneys and the liver, and it tends to cause some dysfunction there. And you could imagine when your liver and your kidneys aren't functioning properly, you're not going to feel well. You're going to be nauseated. You're going to be lethargic. You're just not going to feel right. And so we have these physical symptoms. But we also have psychological symptoms, including hallucinations, and in cases where people are consuming it over and over and over, it can literally lead to almost like a paranoid schizophrenic kind of state. Now, here's the thing. Before it was known that the ergot was toxic, what would happen is the grain would be harvested and ground up into flour. And so many people, throughout history have suffered from ergotism or ergot poisoning. And we actually implicate ergotism, or some scholars do, in some historical events which occurred both in the United States and in Europe, most notably the Salem witch trials. And so, what we believe was happening, we might think a little bit about the history of the Salem witch trials and what happened. Basically, you had the majority of the community, in this case in Salem, Massachusetts, accusing people that were kind of living on the fringes of society of being witches. And they literally rounded them up, and they had these trials. And the trials were interesting because you could admit to being a witch and make your penance with God, which is what they wanted you to do, and then they would put you to death. Or you could deny being a witch and they would put you to death anyway. So there wasn't a lot of choice there. So, what we believe was going on, or what is possible as an explanation for what was going on, why there was this hysteria, is the fact that everyone in the community was probably suffering from ergotism. And so these individuals were feeling ill. They were also hallucinating. They were seeing things that weren't there, and there was a certain amount of paranoia involved. And so, what's the evidence of this? Well, they've actually exhumed bodies from this time period and found the presence of ergot in their bones. And so we know they were suffering from ergotism. We'll never know for sure how much of a role that played. This same scenario played out all over Europe. St. Elmo's fire, very similar situation. And, when people realized that those little fungal components in the grain were toxic, and they were careful to remove them before they ground up the grain, these types of events didn't happen as much, okay? So that's not the end of the story either for the ergot fungus, because fast forward to the 1940s, late 40s, early 50s, and this guy, Albert Hofmann, was hired by a Dutch pharmaceutical company to study the ergot fungus. The 1940s is the advent of heart surgery. It's where they're, cracking people's hearts open and tinkering with them. And what they were actually looking for in the ergot, what this company was looking for, was some sort of a chemical alkaloid that could act as a brake on the heart, that could slow the heart rate down. and they believed that the ergot might have been promising in that respect. So they hire Albert Hofmann. Albert Hofmann is a renowned chemist. He is the master of alkaloid chemistry of the 1940s. And what that means is he's able to take a plant, or in this case, a fungus, and separate all the different chemical alkaloids. So Albert Hofmann begins his work, and he finds a chemical called lysergic acid. And being a chemist, he kind of can't leave well enough alone. And so what he does is he takes this chemical lysergic acid and he starts adding a functional group at different locations to this molecule. And the functional group that he starts adding is a diethylamide group. And so he's tinkering around with the lysergic acid diethylamide. He's putting the diethylamide in different locations on the molecule. He makes 31 different derivatives of this molecule. And one day he's walking across his laboratory carrying a vial of LSD-25, which was the 25th version of the molecule. And Albert Hofmann is admittedly a clumsy man, and he kind of stumbles, and he spills a tiny bit of this liquid onto his wrist. And later he writes, I had this profound psychological experience from this. And he was very surprised because most chemicals aren't readily absorbed through the skin. So for about 6 months, Albert Hofmann, he wants for someone to ingest it to see what it will do. He's pretty sure that it's the lysergic acid that is responsible for the hallucinogenic component of the ergot, but he doesn't know what it will do. He's afraid it might kill someone. And so he finally decides to be the guinea pig. And so Albert Hofmann in his laboratory one afternoon takes what for most chemicals or drugs would be a small amount. He takes about 20 milligrams of pure LSD. And just to be clear, we measure doses of LSD in micrograms or thousandths of a milliliter, and about 20 mics would be considered a pretty strong dose of LSD. So in his lab, Albert Hofmann takes the equivalent of thousands of hits of LSD all at once. And his goal is to journal. He's going to journal every 15 minutes. Well, as you could imagine, things go awry. Albert starts tripping, and he makes it about an hour and a half, and then he realizes he's in trouble. And he for some reason, decides he needs to leave his lab and get back to his flat, his apartment. So he leaves. He's on a bicycle. He's on the big wheeled bicycle of the day, and he doesn't make it. He gets about halfway betwixt his laboratory and his apartment, and he falls off the bike, and he's laying on the sidewalk gawking at people and pointing at them and laughing. And this is one of the most famous chemists in the world at the time, and people are like, Albert Hofmann, he's drunk at 2 p.m. in the afternoon. So luckily for Albert Hofmann, a friend came along and found him on the sidewalk and got him back to his apartment, where he wrote out what must have been this horrific LSD trip. Okay, so kind of a funny story. As you could imagine, Albert Hofmann is a cult hero. Here he is holding the LSD molecule. And here's the crazy thing. Albert Hofmann lived to be 102 years old before he died. And so it wasn't that long ago that he died. Of course, they had a big party for him on his 100th birthday. And, someone had to ask him, do you attribute that giant dose of LSD that you took to your longevity? And Albert just kind of shook his head and laughed. So we'll never know for sure what he thought about it. It's safe to say that this discovery changed human culture, right? With the discovery of LSD, we had, the 50s and into the 60s with the acid tests and Ken Kesey and, all the history that goes along with that. LSD is still used as a psychoactive drug. It is, we could talk a little bit about LSD in terms of its nature and what it does and how it affects. Just making sure I'm not leaving anything out about Albert Hofmann. Albert Hofmann teamed up with a very famous ethnobotanist named Richard Evans Schultz. And between the two of them, they discovered ayahuasca and many other psychoactive alkaloids. Schultz was an ethnobotanist and a linguist, and he would go into South America and Africa and learn the language of the natives. And then he would take the different ayahuascas or different analogs of psychoactive chemicals that were indigenous in that tribe. And then he would send those plants back to Albert Hofmann, who would separate the alkaloids and discover things like dimethyltryptamine or DMT. Okay. So what about LSD? Some myths and some truths. First thing I'll tell you is don't think you can eat ergot to get high, because ergot is toxic. The lysergic acid diethylamide, LSD, is a semi-synthetic drug. It's been altered in a lab, okay? It is completely water soluble. Here's a crazy thing. You smoke a joint, 6 months later, they can pluck a hair out of your head. They, I guess, being the man, can pluck a hair follicle out of your head, and 6 months later they can detect THC in your system from the hair. 48 hours, really almost you could say 24 to 48 hours after ingesting LSD, it is undetectable because it's water soluble. You pee it out, you get rid of it rather quickly. Okay. Because it's water soluble, there's always been some mystery around the flashback, and we might talk about that in a bit. So what is it and how does it work? It is a serotonin analog. Serotonin, not spelling very well, sorry, let me do better. Let me try that again. Serotonin is a brain neurotransmitter. It's also called hydroxytryptamine. There are many chemicals which mimic serotonin, including LSD, DMT, dimethyltryptamine is very similar to hydroxytryptamine, psilocybin, and many others. These chemicals, your brain can't really tell them apart from serotonin. And so when you take these different hallucinogenic or psychoactive chemicals, you're overloading your brain with neurotransmitters. And this is what accounts for the hallucinogenic effect of these drugs. Okay. Some crazy things about LSD, there's no lethal dose, meaning you could literally, drink a vial of it or a glass of it and it wouldn't kill you. However, you might wish you were dead. And it's not to say that people haven't died while under the influence of LSD, because they can do really stupid things when you get that high off of it. Okay. So one of the phenomena that's been known for a long time about LSD is that of the flashback and how to explain this. It used to be believed that somehow LSD was stored in your body, in your cerebrospinal fluid or something like that. This has all been debunked. There's no evidence that the LSD is still in your body when you have a flashback. The discovery and what they think is the explanation for the flashback is that when you overload your synaptic network with a serotonin analog, it creates literally new synaptic patterns in your brain. And this is kind of a wacky idea, but it's kind of like when you remember something. In order for you to remember something, a specific pattern of neurons in your brain has to fire. When something's on the tip of your tongue, part of that pathway is firing, right? Well, what they believe happens is when you take LSD, it creates these new synaptic pathways. And then for whatever reason, usually there's some trigger, these synaptic pathways will fire at a later date, usually days or weeks later, and you have that temporary feeling like you're under the influence of the drug again. Okay, so this is some pretty wild stuff. Now, there has been some speculation about the human brain and the effects of these drugs. There's one theory that suggests what accounts for the explosion in the size of the human cerebrum compared to, say, prehistoric humans like Cro-Magnon, is the ingestion of hallucinogenic chemicals and the overstimulation of the brain causing synaptic patterns to form. Okay, it's a theory. All right. So that's a pretty good quick and dirty about these different chemicals. And we will move along to the next topic, which is a rather sad topic and has to do with members of the same group, the Ascomycota, which are responsible for causing what we call blight in plants. And one example is, this is a funny word, Phytophthora infestans, which is commonly referred to as the Irish potato famine. Okay, this particular fungus is what caused the Irish potato famine. So still around, and it's often referred to as late blight. You can see here it is on potato, here it is on tomato. If you grow tomatoes in Western North Carolina 2 or 3 years in a row, eventually you're going to see these lesions, these black lesions of the late blight. Even commercial non-organic growers have problems with late blight. It's very difficult to control, even with systemic fungicides. And eventually it will come and reach the North Carolina or the US. It can overwinter in the soil, but it more commonly comes on potatoes that are blighted, or even tomatoes that are blighted, that are brought from tropical areas into the US during our growing season. Okay, so a little bit about the history of Ireland and the Irish potato famine. The Irish potato is not native to Europe, right? The Irish potato is actually native to the Andes, specifically Peru and Bolivia, where these countries come together. When the conquistadors arrived, the indigenos that lived in these regions had 1,000 different varieties of potato. This was the staple crop of these people. And, you know, they had a potato that would grow all the way from the ocean all the way up to 12,000, 15,000 feet in elevation. The potato is in the nightshade family. And when it was brought back to Europe, the Europeans scoffed at it as being a food because so many of the European nightshades, like belladonna and mandruga, are very toxic. And so the Europeans were like, we're not eating this thing. There's no way. It was actually, there was a French aristocrat who saw the importance or the potential importance of the potato, and he decided to plant, he leased an acre of land right outside of Paris, and he planted an acre of potatoes out there. Well, the peasants of France who were starving were like, hell yeah, we'll eat these things. And so they stole some potatoes from this field and began planting them themselves. Okay. So the potato really became established in France and then eventually in Ireland. And the thing to think about Ireland is it's a big rock. There's not a lot of really deep, fertile soil. And so the potato, prior to the potato, the Irish were basically surviving on turnips and milk or buttermilk to get through the cold Irish winter. Okay. With the introduction of the potato, now they have this very nutritious staple crop. And just to give you an idea, prior to the potato, there were about 3 million people living in Ireland. Over the next 100 years, when the potato was introduced, went to 12 million. And it's said, you know, the potato was the direct reason why this happened. Potatoes are very nutritious, particularly if you eat the skin. You can actually survive for a very, very long time eating nothing but potatoes. There's just enough vitamin C in the skin and potassium and micronutrients, and certainly plenty of carbohydrates to live a long time, okay? So, you know, the Irish really loved their potatoes. Well, everything was rolling along until the 1860s, when they had a particularly wet summer in Ireland, which for Ireland, you can imagine that must have been very wet. And so they harvested potatoes just like they had done for 70 or 80 years prior, and they put them in storage and they all rotted. And what had happened was they had been infected by this fungus, Phytophthora infestans. And so we had a complete crop failure after harvest. And you have 12 million people depending upon this one crop to survive the winter. And so what happened? Well, mass starvation, mass famine, right? And, you know, the second year, many people died the first year. The people that survived, they grew and harvested their potatoes the next year, and the same thing happened. We had 2 years in a row of crop failure. At that point, we had millions of people starving to death. Now, there's always been a rift between the English and the Irish, and one of the reasons has to do with the fact that the English that grew large, copious amounts of grain, particularly wheat and barley, had enough food to feed all of England plus all of Ireland. But at the time, there were tariffs in place, and it made it, or actually not even tariffs, but laws that made it illegal to export food to Ireland from England. It was simply against the law. By the time a few good Samaritans worked to get these laws changed, it was too late, and the Irish had died. So how many died? 3 million died of starvation. Imagine that, 3 million. 3 million left. A third of the population starved, and a third left. And where did they go? Most of them came to the New World to make a better life, right? So if you are of Irish descent and you are in the United States watching this video, there's a pretty good chance that you're sitting here because of a fungus, right? And so this is a really dramatic example of ethnobotany and the relevance of both plants and fungi in this case, because they're both involved, right? You know, it's fascinating story. We do, you know, have a lot of history in relation to plants and fungi as well, okay? So there have been other fungal blights, unfortunately, within the Ascomycota, and most notably in this country is the American chestnut blight. The fungus responsible is Cryphonectria parasitica, and a little bit about the history of this because it's also a fascinating story. The American chestnut was the dominant species in Western North Carolina, which was sort of the center of its range, all the way to north, all the way into New York State, and south all the way really into Alabama and Georgia. So, you know, we're talking about probably safely to say billions of trees. In Western North Carolina, every other tree, and in some cases every tree, was an American chestnut in the forest. The forest was dominated by these American chestnuts. And, you know, so what happened to them? They're gone, effectively. And as the story goes, in 1907, in New York State, a nursery began importing Chinese chestnuts into the United States. As an orchard tree, the Chinese chestnut is relatively small, about the size of a standard apple tree, and they became popular in people that had small yards, with people that had small yards. Well, unknown to anyone, these Chinese chestnuts had a fungus associated with them, and they had evolved with this fungus, and they were quite resistant to it. So it didn't kill them outright, it just infected them. Well, there's this thing called species resistance. And when you evolve with a species, you over time develop, if it's a parasite, you either die or you develop a resistance, or some individuals develop a resistance, and that's what we had. But since the American chestnut had never been exposed it had 0 species resistance. And once this fungus made the leap from the Chinese chestnut to the American chestnut, it was a wholesale death of the American chestnut. So we'll talk more about that in a second. We might, first of all, talk a little bit about why the chestnut was so important. Durable, soft compared to most hardwoods, and rot resistant wood. There is no wood that we know of that is more resistant, certainly not in North America, than the American chestnut. It was used for everything from railroad ties to telephone poles to lawn furniture to log cabins, you name it. It was soft, and so it was easy to carve, yet incredibly rot resistant. Okay. What about the nuts? Mast, right? Anytime we have a crop of nuts that trees make, we call it mast. And the thing about the chestnuts is they're among the most incredibly reliable mast producers on earth. They're the most reliable, meaning every single year they would make a huge crop of nuts. And you could imagine a tree this big would literally make metric tons of chestnuts, and so they were so plentiful. These were eaten by humans because they're very nutritious, but also by wild animals and also by livestock. So back in the day, you didn't have to worry about what you were going to feed your pigs and your cows in the winter. You just let them loose into the woods and they would get fat on chestnuts all winter. So these two things made it a very important plant or tree, especially in the Southeast. Well, when the fungus hit, it spread from New York State, from its center point in all directions along the range of the chestnut. It's believed that it spread about 20 miles a year. It eventually reached Western North Carolina in the 1930s, late 1930s, 1939, and by 1943, all of the chestnuts were gone. So you could imagine millions and millions of trees dead for 20 years after the mountainsides were brown. Imagine all the mountains brown year round. That's what it looked like. Now, here's a really sad thing. In anticipation of knowing that this blight was coming, many of the people in this area prematurely stripped the bark from these giant chestnuts. They were after the tannins, which are used and were used back then to tan leather. And the tannins were valuable, and they thought, well, the tree's going to die anyway. We're going to go ahead and strip this bark off so we can get the tannins and make a little money. Well, this was a very short-sighted approach. Probably, theoretically, some of these trees would have been resistant to the fungus, but we'll never know because they were killed prematurely. And so, unfortunately, we lost these trees. Now, really, since the 40s, they have been breeding the American chestnut, or what was left of it, with Chinese chestnuts in an effort to make a hybrid that is resistant to the fungus. The idea is you want to eventually be 99% American, and then in that 1%, you have the resistance gene. Well, they've been working on it. They're up to about 7/8 right now. It's very time-consuming and painstaking process. It can take 7 to 10 years for the tree to make nuts. And so it takes 7 to 10 years to get the next generation. And so you could imagine it's very difficult to do this. Well, a biogeneticist in Japan has now created a GMO American chestnut. Game changer, right? Because what he did was, he's like, this is too time consuming. I'm just going to find the gene on the Chinese chestnut that gives its resistance to the fungus, and I'm going to insert that gene in American chestnut. And that's what he did. And now he's cloning that times millions. And so theoretically, we could release this GMO back into nature. and it would be 99.9999999 percent American chestnut. And in that tiny little bit that's Chinese is the gene that gives it resistance. I don't know, this, if there's a GMO I can get behind, this might be it. Okay. All right. Well, more recently, we've had Dutch elm disease, which killed all our American elms. Same kind of scenario. Some elms from Europe were introduced and there was a fungus on them that killed our American elms. And then most recently, dogwood anthracnose is killing our native dogwoods. And so, you know, it's difficult to see the fungus when it's in its spore stage. And so we can't always screen or quarantine for these things, although we now try. Okay. I think that is it for the Ascomycota. Let me just go to the next slide and see. Yeah, and so that brings us into the Basidiomycota. And what we're going to do, we will revisit the Ascomycota a few times. Well, before we move on, one of the things that we must say is, you know, we must say, what about the yeasts, right? They're ascomycetes. And so we would be remiss if we didn't mention the yeasts. So we'll go back to this slide for just a second. Talk a little bit about the yeast. Why are the yeasts important? Well, think about it. What do we make with them? Alcohol, right? We make leavened bread as well, but alcohol would be the most important byproduct of yeast. And so, you know, how was alcohol invented? It wasn't invented, it was discovered. Most likely someone put some fruit or fruit juice in an animal bladder and the wild yeast fermented it. And we know all about the fermentation process. And, you know, Joe and Sue caveman and cave woman drank it and they were like, whoa, right? So there's that. And then, you know, we also know that yeast can be a parasite. There's, you know, a yeast infection, or ringworm is a good example of a yeast that can become infective of humans. Candida is another one. You can get a yeast infection from Candida. Okay. So the yeasts are hugely important, and they are distinct as fungi because they are unicellular. They are a single cell. Okay. All right, I think we can go to the next slide now. So we'll just do a little intro to the Basidiomycota. We will revisit the Ascomycota when we talk about with some symbiotic relationships, specifically lichens. Okay. And we'll also talk about mycorrhizae, which can be formed with Asco or Basidiomycetes, which is another symbiotic relationship. We're going to do those at the very end of our discussion of these groups. Okay. So what about this next group? Well, as you can see, it is the largest and most diverse group. How is it relevant? It includes many poisonous species and many edible species. It includes the most poisonous species of fungi, which incidentally are some of the most poisonous things on Earth in terms of, you know, the amount of the chemical that it takes to kill a human. Okay. It also includes many psychoactive species, including psilocybe. The genus psilocybe are the so-called magic mushrooms, which contain psilocybin, which is another serotonin or hydroxytryptamine analog. Okay. It also includes Amanita muscaria, the fly agaric, which has a different set of chemical alkaloids that can induce a semi-comatose state. Okay. Some common names: mushrooms, puffballs, shelf fungi, stinkhorns, coral fungi are just a few of the sort of common names. Gill fungi might be another way you could think of this group. Okay. I'll tell you that the structure, and we're going to do the life cycle in detail, but I'll tell you that the structure that's formed during sexual reproduction that makes it in the Basidiomycota, as you might guess, is called a basidium. And so you're going to see this word again. Okay. I think we'll stop there. It's a pretty good place to stop. So what we have left to do in this unit on fungi is talk about the Basidiomycota and the Deuteromycota. And then we'll come back and talk about a few things that are kind of on the fringes, like slime molds and water molds, and then some symbiotic relationships between the fungi and some other organisms. Okay. All right, team. Well, had another fun day with you. I hope you're doing well, and I'll see you next time.